Hello, my name is Travis Monk. This is one of a series of videos on cell biology. In this video, I will describe how to properly use a compound light microscope in a lab setting. There are many different types of microscopes out there. Dissection microscopes, as shown on the left, are used like a magnifying glass to look at the surface of something that's usually visible to the naked eye. You might use this type of microscope, for example, to look at the eye color of a fruit fly. Electron microscopes, as shown in the image on the right, work by using electrons to precisely map the surface of an object. These microscopes can be so large that they fill an entire room. They can cost tens to hundreds of thousands of dollars and are typically only found in universities or research facilities. Again, the emphasis of this video will be on the use of a compound light microscope. An example of this type of microscope is exhibited in the middle picture. Compound light microscopes are the most commonly type of microscope used in a high school science classroom setting. Compound light microscopes are typically used to view objects that would be impossible to see with the naked eye. Light microscopes work very differently than dissection microscopes that were described on the introductory slide. Instead of viewing the surface of an object, as in a dissection microscope, a compound light microscope works by passing light through an object to make it visible. If you were to try to look at a large object, such as a finger, under a compound light microscope, you would not be able to see anything at all because not enough light would be able to pass through. To view things using a compound light microscope, you need incredibly thin segments of an object, such as cells. These thin segments are typically placed on a glass slide. To properly use a compound light microscope, you need to be familiar with many of the parts and what they do. To avoid communication problems, it's important that you know their proper names. If you were to ask your teacher what to do to make the object that you're looking at clear, an answer such as turn that thing on the side of the microscope wouldn't do you a whole lot of good. When you first begin using a microscope, you'll probably need to get the microscope from the microscope cabinet. Since these microscopes are very expensive pieces of equipment, it's important that you take proper care of them. When you move the microscope from one location to another, it's important that you hold the microscope with one hand on the bottom or the base of the microscope and another hand on the arm of the microscope, which holds all the other pieces together. By doing this, you're much less likely to drop and damage the microscope. With compound light microscopes, you're almost always going to use slides to view whatever you're interested in looking at. Sometimes you will just use already prepared slides, and sometimes you'll need to make these slides yourself. Once you have obtained a slide, however, what you need to do to view the contents of the slide is place it properly on the microscope. Your slide should be placed on the stage of the microscope. It's called a stage because it holds and exhibits whatever you want to look at, just like you would watch a play on a stage. Light passes through a small portion on the stage of the microscope. Placing whatever you want to look at above that hole that light passes through will allow you to illuminate the object. You should secure your slide on the microscope using stage clips. They will keep the slide in place, preventing it from moving around too much when you don't want them to. While held down by the stage clips, you should be able to manipulate the slides carefully so that you can find whatever it is you're looking for. When you have your slide in place, you should turn on the light source. Since compound light microscopes work by passing light through an object, you need to make sure that you have plenty of light available. You plug most modern microscopes in and should find a light switch on the back of the microscope. Once you've turned your microscope on, look through the eyepiece to see what is on your slide. Your eyepiece not only allows you to view the slide, but it also magnifies what you're looking at 10 times. The chances are that when looking through your microscope, you're not going to find exactly what you want right away, especially if your microscope is magnifying what you're looking at too much. You can choose how much your microscope is magnifying whatever you're looking at by moving the revolving nose piece. The revolving nose piece is connected to 3 or 4 different objective lenses on most microscopes. On our microscopes, the shortest lens is referred to as the scanning lens and is colored red. The scanning objective lens magnifies whatever you're looking at 4 times. If you take into consideration the 10 times magnification from the eyepiece, there's a total magnification of 40 times. You'll always want to start looking at things underneath the microscope using the scanning power objective lens, as it makes it easier to find what you're looking for. After you've found and focused on whatever you're looking for with the scanning lens, a process that will be described in the next few slides, you can move it to the next largest and next most powerful lens called the low power objective lens. The low power objective lens is typically colored yellow and magnifies what you're looking at 10 times. Taking again into account the 10 times multiplication of the eyepiece, the total magnification using the low power objective lens is 100 times. If you want to get an even closer look at your slide, you can switch to the high power objective lens, which is typically colored in blue. This lens is larger in size yet and magnifies 40 times. Again, with the eyepiece, the total magnification is 400 times. Using this lens, you can see even the smallest types of cells, or prokaryotes. One of the most difficult things to do with a microscope is get whatever you're looking at into clear focus. There are two knobs on the arm of the microscope that you can use to get things in focus: the coarse and fine adjustments. The coarse adjustment is the larger of these two knobs. It's called coarse because it very roughly puts things into focus. You should only use the coarse adjustment when you're viewing things under the scanning, or red, or low power, yellow, objective lenses. The smaller of the two knobs, the fine adjustment, should only be used under the high power, or blue objective lens. You shouldn't use the coarse adjustment while under high magnification, as you could damage the slides or the microscope itself. When trying to put slides into focus, it's very important that you start with the scanning power objective lens and get things into focus, and then move to the low power objective and again put things in focus before moving to the high power objective lens. You'll have a lot more success and a lot less frustration doing this than jumping right to the high power objective lens. The reason for this is exhibited in the picture on this slide. If you're using the red scanning objective lens, you can see a good deal of the slide that's supposed to be represented in white. Effectively, you're not zooming in on your subject too much. If you're slightly off center, you might still see some of the cells or whatever subject you're looking at for. In addition, getting objects into focus using the scanning power objective lens is comparatively easy. The yellow, low power objective lens is more than twice as magnified as the scanning lens. As a result, any errors that you make get magnified. If you're slightly off center, you might not find anything in the field of view. In addition, you need to be a lot more precise when you are trying to get things in focus using the coarse adjustment. Using the blue high power objective lens is 4 times as magnified and probably 4 times as difficult as using the low power objective lens. You're looking at a much smaller segment of the slide, again as exhibited in this picture, and the probability of being off center is pretty likely. One overcorrection with the coarse adjustment and you could completely lose whatever you were just observing seconds ago. Trying to find and then focus on your subject with the high power objective lens without using the scanning or low powers is like looking for a needle in a haystack. If you're ever looking at something underneath a microscope under low or high power objective lenses and then for some reason things get out of focus or you get lost, the best bet is to go back to the scanning power objective lens and put things into focus again. The last part of the microscope that you may need to use from time to time is referred to as the diaphragm. As I mentioned earlier, light passes through a small hole on the stage of the microscope. The size of that hole can be enlarged or made smaller by manipulating the diaphragm. The diaphragm often looks different from microscope to microscope, but it will almost always be found below the stage of the microscope. If what you're looking for under the microscope is too bright or too dark to see things properly, you can try to adjust the diaphragm. That is the end of this video on how to properly use a compound light microscope. If you're interested in learning more about cell biology or any other themes of biology, please subscribe to my channel. Thank you.